Good morning, everyone. Thanks so much for coming. My name is Dr. Christoph Meinert, co-founder, CEO of Gelomics. So look, today I have a little bit of a presentation introducing you to Gelomics, also showing you in high level some of the applications of our first product series, which we've commercialized around 2 years ago. And lastly, as Norbert pointed out, I'll also discuss a little bit career pathways, opportunities to engage and collaborate with us if you have interest of that. And please, I would like this to be as interactive as possible. So if you have any questions, anything like that, feel free to just shoot the question and I will address those as well. Okay, so just a little bit of background, and I hope everyone online can hear me as well, which I believe they do from our testing earlier. So Gelomics is a startup company based in Brisbane. We're based in Kelvin Grove, and as Norbert pointed out, we came out of QT research. So the company was incorporated in 2018, so already quite a while ago. But the first couple of years, we've really focused on developing what we refer to as a minimal viable product. So in the early couple of years, all the founders had full-time jobs, and we did Gelomics essentially on the side. Right, so we then worked our way through developing basically what we can see here on the table today, a very simple product that is, however, highly effective as well. And we've commercialized this. We soft launched it approximately 2.5 years ago now. And we've been able to very quickly grow the company and also embed the products and research practices all around the world. And really what we're trying to do is change the way that drug development and research and development in particular is performed. And I'll talk about this in a bit more detail on a later slide as well. So our technology essentially allows cells to grow in a three-dimensional environment to form small microscopic representations of human tissues. So it's a tissue engineering technology in the in vitro space as well. And the idea is that we can accelerate preclinical drug development, for example, by reducing reliance on animal testing and making it more efficient, more effective as well. So I've already touched on sorry, I'm always on the wrong computer here. A little bit on the background. And since we've soft launched our product, it was really we really heavily focused on validating the core technology. And we've been able to do that over the last 2.5 years or so. So our typical model of how we engage with our partners and customers is that we try to get them to do a demo. And of those demos, over 80% have now converted to sales and also ongoing partnerships. So now we have over 300 academic customers across 23 countries internationally. So it's growing pretty quickly. We've just also growing our global distribution network. And small world, we actually signed an agreement with Norbert's brother in Switzerland, who is distributing our products in Switzerland. Yeah, I actually just found out today that Norbert is the brother of the person we talked to in Switzerland. And yeah, we've bootstrapped for a long time, meaning we've basically survived and grew the company, developed our first products on very lean and little capital. Basically, founders invested a bit of money into it, and we all kind of worked for free on it and did a bit of work. So that allowed us to generate some early revenue and also de-risk the technology, show that to investors that this is really something worth pursuing. And only last year in December, we've raised our first external venture capital round, which was a $3.2 million pre-seed round. And at the time, in November last year, we had 2 employees. Now we're close to 20. So we've grown quite quickly. And we've also been able to secure the first partnerships with large pharmaceutical companies. So we're now working with Boehringer Ingelheim and also AstraZeneca on a couple of drug development projects as well, which is, for us a major milestone, of course. So the problem we are addressing, and I think I've touched on that a little bit as well, is the inefficiencies in preclinical drug development. So we all know that cell and animal experiments still form a very integral part to preclinical drug development. So in 2021 alone, it's estimated over 50 million cell culture experiments were performed, and close to 200 million animals were used for research purposes. Unfortunately, these models rarely actually predict human biology and human drug responses accurately. So in fact, over 90% of drug candidates that successfully pass through both cell and animal studies fail when tested in human clinical trials. Clearly, it doesn't really translate very well, right? And there's an associated burden on the pharmaceutical industry in particular, because the failure of a single drug candidate when it fails in clinical trials typically costs around 1.3 to 1.5 billion US$ per unsuccessful drug. With literally hundreds of drugs failing every year, you can imagine the expenses the industry has to cover is massive. And we're trying to change this by having better model systems using our LunaGel technology. So essentially, this technology allows you to grow small representations of simplified human tissue models in a very reproducible manner. I just realized I have a timing on those slides, so apologies if they skip. I forgot to turn that off. Maybe I'll have to just quickly fix that. Sorry, everyone. Will just be a second. Use timings. Here we go. And we use a photo cross-linking technology, and I'll introduce that to you a little bit on the next slides as well. And the idea is really that we can, create very simple and reproducible 3D cell culture models that are more closely resembling human biology than traditional cell-based methods or even animal experiments. Of course, by no way we can fully replace animal testing right now, but in many cases we can aspects of it. So an example is that our pharmaceutical partners that we work with, they work on, immuno-oncology projects, and they basically all of their work is done in mice. And they report that the take rate, for example, for the experiments is around 50%, meaning that half of the mice are basically not creating the data sets they are after. The experiments can take many months, 2 to 3 months. They're expensive. They require, facilities for the mice, very trained and experienced users, and so on. And many of these things that they're actually trying to study, we can perform and study in a much more simplified format in vitro, where you actually get results in a week or two instead of 2 or 3 months. The costs are literally around 10% of animal testing, and you actually get more mechanistic insights in many ways as well. Of course, again, you cannot replicate the full environment that we see in vivo, but we can do certainly parts of it. And what we do and how our products work is that we use natural biomaterials like gelatin, hyaluronic acid, and so on, and we chemically modify those. And the benefit of this is that these materials are usually manufactured under pharmaceutical grade conditions at multi-ton batch scale. So they're very reproducible, very well controlled, and at the same time also offer the biological activity that is needed for cells to behave in a more physiological manner. Meaning the cells can interact with the materials through different cell surface receptors that then trigger intracellular signaling and help the cell to develop their native phenotypes. Also allows the cells, of course, to interact with each other, which is a very important feature as well. And in many cases, depending on the specific matrix, is also responsive to proteolytic degradation. That's, again, important. The remodeling process is important because it allows cells to migrate through it. It allows cells to deposit their own matrix within a 3D environment, and so on. And by adding this chemical moiety that allows us to photo cross-link the system, so meaning you go from a solution to a gel-like state by exposure to light, that allows you to highly control some of the key properties that in turn also influence cell response, such as the stiffness, the porosity of the materials, and some other factors as well. And this is the general workflow of how the products work. So we really put emphasis on simplifying the process. And that's really comes from our market research, particularly of users for Matrigel. So if you ever worked with Matrigel, you know it can be quite a pain to work with, right? You need to thaw it in the fridge overnight. You have relatively viscous solutions. They need to be kept on ice. You need to keep your pipette tips on ice, your cultureware, everything. And also the gelation process of actually making the gels, is a temperature-sensitive process that's driven by collagen fibrillogenesis, which in itself is very sensitive. It's sensitive to pH, to concentration, biochemical composition, the temperature, many factors that influence it. And ultimately, the gels that are resulting from it are poorly controlled in that sense that the mechanical properties can be different, between experimental replicates, between batches, and so on. And we try to really simplify this. So our products really come as a ready-to-use kit. You simply mix in your cells into the solution. You dispense it into well plates or any other type of culture formats, even microfluidic chips. It then moves into this little, very basic photo cross-linking device, which has a visible light LED panel inside of it. It's a blue light, so it's cytocompatible. And it shines light onto the samples, the materials. They form a covalently cross-linked polymer network in which then the cells grow and develop into 3D cell cultures. And one of the important things that we can tune and tailor reproducibly is, of course, the stiffness of the material. And that's quite important because different cell types in the human body are actually intrinsically tuned to the native microenvironment they originate from. So with other words, a neuronal cell, a brain cell, typically sits in a soft and squishy microenvironment, the brain, right? So it's actually critically important for the cells when they're cultured in vitro to also replicate these mechanical properties for them to allow them to basically develop their physiological structures, neuronal networks, and also even gene expression and other phenotypic expressions as well. On the other hand of the spectrum, you might have, cartilage tissues or bone tissues, which of course are very, very stiff. And again, the resident cells are also tuned to the intrinsic properties of that matrix. And as many of you know, diseases can also change the material properties of the extracellular matrix. Many cancers, for example, the extracellular matrix increases in stiffness and the collagen fibrils get thicker over time. And this in turn, both in vitro experiments, but also clinically has been shown to increase the rate of metastatic processes, EMT, and so on. So it's really quite important to be able to replicate this in vitro to study the mechanistic insights underlying these processes. But if we look at market-leading products such as LunaGel, it's very difficult to control the mechanical properties because, again, of all of the factors I mentioned earlier. And typically, they're much softer than real tissues would be. So it's difficult to achieve physiological behaviors, of course. Our kits always come either as a low stiffness or high stiffness kits to cover a very large range of mechanical properties, covering most of the common cell types. And by far, I would say the main product sold is the low stiffness kits, which are for like fibroblasts or cancer cells, organoids, anything like that. And the way the mechanical properties are controlled is by simply adjusting the duration of light exposure in our photocross-linking device. So here you can see two graphs that are representative for one single biomaterial that we are selling, which is the bovine bone gelatin-derived LunaGel. And you can see the relationship between cross-linking time, so that's the light exposure duration, and the mechanical stiffness here expressed as the compressive modulus. So I think the key takeaways here is that there's a relationship between the two. You can tailor it to your requirements. And the entire process takes an absolute maximum of 8 minutes. So most cell types that we work with and others work with, they would usually require cross-linking for only 2 to 3 minutes. So when you look at LunaGel, you're looking typically a 45-minute or so protocol for making gels. Here you can cut it down to 2 minutes. And there's no requirements for keeping the solution cool or anything like that. They're also quite low in viscosity as well. And controlling the stiffness, of course, does influence the phenotype. Here's just some morphological images of different cell types as a representative, where you can see that, for example, the spheroid size of these MCF7 spheroids is inversely correlated to the stiffness of the material. And likewise, you can see these MSCs, these are bone marrow-derived human MSCs. And depending on what type of microenvironment they're cultured in, you can see there's clear morphological differences. And we don't have the data here in the slide set, but we can also definitely see that if you culture them in stiffer microenvironments, they tend to differentiate towards an osteogenic or chondrogenic lineage as well, which is a very interesting finding. And my plan for today is to kind of just highlight a couple of applications going from very soft to more stiffer hydrogels. And again, this is relatively high-level scientific data, so I won't go too much into the details. If you have any specific questions, please feel free to reach out. So first thing I wanted to touch base on is capillary system. Yes, please.
Sorry. Stiffness control. How does that correlate with
Yeah, so it is directly correlated as well. So typically, as you expose longer, you also create a smaller porosity. Unfortunately, it cannot be independently controlled. Yeah, so it goes hand in hand. Yeah, so.
What happens if you expose it longer than 8 minutes?
Look, there's a maximum. There's basically how the system works is imagine there's a molecule inside of the mixture that can absorb the light and initiate the photocross-linking reaction. It's called a photoinitiator. And that photoinitiator essentially acts like sunscreen for the cells. So that means it absorbs the light that comes in and reacts off, essentially, before the energy of the light can hit the cells. So that means, with other words, that if you exhaust the entire photoinitiator that's in the system, then you will also have effects on your cells. So that's typically after maybe 15 or so minutes of exposure. That's when the photoinitiator is depleted. And then you can also potentially impact viability of your cells.
But then you're not impacting the stiffness of the gel anymore.
No. Exactly, yeah. 8 minutes, 8.5 is basically reached maximum for this particular. Yeah. No, it's not. I mean, if you put it in the sun, yes, But in a laboratory environment, typical LED or even like the fluorescent bulbs, they don't contain the wavelengths in a significant amount of intensity to cause curing. So you can easily leave it out under light, that's no problem. But yeah, very good questions. So yeah, capillary system. So capillaries, I think everyone is familiar with it, right? In the human body, except for in cartilage, every single cell in the human body is like within 50 to 100 micrometers of a capillary. Capillaries are the small, tiny vessels where basically all of the exchange of nutrients, oxygen, and waste products occurs, right? So it's critically important to actually grow capillaries in vivo or in vitro, for example, for tissue engineering applications. So, you cannot really grow an organ or a tissue if you can't vascularize it. So, and it's a key thing that many groups have been working on to try and generate these capillary systems in vitro. And again, this is on the softer end of the spectrum in terms of mechanical properties. And here are just some examples of how the cross-linking duration influences capillary formation, capillary-like network formation by HUVECs, so human umbilical vein endothelial cells, and MSCs, which act as parasites. They basically support the network structure. And you can see there's a clear relationship between, the cross-linking time or the stiffness of the material and how those capillary systems form. So by day 6, this is an optimal condition here. You can see there's very extensive networks of cells forming that express also CD31. That's a vascular or angiogenic marker. And then it's a little bit small, but maybe you can appreciate there's some like red cells that are attaching or sitting on the vascular part, the green cells, and these are essentially parasite-like cells, a subpopulation of the MSCs we add to this, and they stabilize the network and make it stable for at least 4 weeks. And that's another key advantage. So in Matrigel, you can sometimes, depending on the batch, generate vascular structures like this, but they're typically, because the mechanical properties of the system are much lower, they fall apart, they disintegrate within a few days. In our system, you can culture it for a long duration of time, and that actually allows you to do pretty cool things. So if you grow these, for example, on a microfluidic device, and here this is an AIM Biotech commercial chip, you can also generate these network-like structures. And if you then start perfusing the system, the capillaries will actually mature into perfusible capillaries. And hopefully you can see this on here. So these are not stained, unfortunately, but you can maybe appreciate on the left-hand side, we've got little small particles. You can see they are actually perfusing all the way through the capillary structures which are formed by the cells. So using a system like this, you can imagine you can study things like metastatic processes, uptake of, let's say, molecules or drugs into a vascular system, or these type of processes as well. Blood-brain barrier models are a very common application of using these cells in combination with other cells as well. That was on the softer end. So now let's move a little bit higher in the stiffness. And one example, or a couple of examples I want to talk to you about is oncology and cancer research in general. So I already touched on this a little bit in the introduction part, that the stiffness of the ECM typically increases with the progression of cancer, right? That's well described for prostate cancer, breast cancer, and so on. And I wanted to show you here that we can actually replicate this, again, in a very simple in vitro format. The images you can appreciate here are MDA MB231 breast cancer cells. So these are metastatic breast cancer cells. And we essentially preformed spheroids by simple aggregation. And then we plated those spheroids within ECMs of different mechanical stiffness. On the left-hand side, we have a system that replicates more healthy tissue, and here, of course, it becomes more stiff, replicating more cancerous tissues. And if you look at the images, you can clearly see that as the stiffness increases of the system, you see more and more cells leaving that initial spheroid, that initial tumor, so to say, and starting to metastasize and spread away from the system. So again, replicating in a very simple format, what we can also observe in vivo. Other studies that we've performed as a proof of concept is, of course, chemotherapy response studies. And here I would like to highlight, again, on the left-hand side here, these are MCF7s up the top row, which are, I guess, a cell type more representative of the primary tumor, a breast cancer tumor. And on the bottom, we have MDA MB231s cultured as individual cells within the LunaGel materials. And if you treat those cells now with Abraxane, right, and you have them in monolayer, you would expect, you would basically look at the relative metabolic activity as a measure of how effective the Abraxane is killing the cells. And you can see that in both cases, it kills around 75% of cells of MCF7s and also the MD8 and B231s. But if you culture these cells in a more representative environment in LunaGel, they form these more representative structures, and suddenly, particularly the MCF7s, become much more resistant to chemotherapeutic treatment, while the killing of the MD8 and B231s, which are the metastatic cells, is more effective. And that's, interestingly enough, this is very well correlating with clinical data sets. So for those of you who don't know, Abraxane in the clinic, through clinical studies has been shown to be effective against metastatic cancers, but not so much for the primary tumor treatments. And this is, again, looking at these in vitro data sets, they correlate quite well with the clinical responses that are seen as well. So in Australia, Abraxane is only used for metastatic breast cancer, or in other cancers as well. Of course, you can also use the system to deliver cells inside an in vivo host. So while we're trying to reduce, of course, the requirements for animal testing, one way of how our materials can be used is to actually encapsulate cells within defined microenvironments and then deliver those cells inside a hydrogel into an animal. So you can also, again, study things like metastatic processes as a function of mechanical stiffness, let's say, but in an in vivo host. How do you deliver them? So there's different ways. We have an injectable system as well that is based, that uses what's called a click chemistry. So you have two materials, you inject them through a syringe and they form spontaneously a gel. Or you can use the traditional LunaGel, pre-cross-link them and then implant them subcutaneously, for example. More of a surgical procedure, yeah. And that click system that we have, that is also used in pharmaceutical companies as well. So they use it now to deliver the cells in vivo as well. Another cool example I wanted to show you, which I thought was really, really nice application of the technology, is the formation of skeletal muscle tissues in vitro. And this is work by James Armstrong. At the time, he was a postdoc in Molly Stevens' group in Imperial College. I met him there. I was there for a couple of months. And we started working together on this. This was published also in Advanced Materials quite a while ago, actually. And James came up with the cool idea of using standing ultrasound waves to pattern cells in a three-dimensional construct. So how that works is you have essentially a Petri dish where you have a gel solution, a pre-gel solution before it forms a gel. You have cells suspended in there. The cells can float around, and by applying standing ultrasound waves, you apply a very tiny force onto the cells that makes the cells essentially move into predefined positions where the force is zero. So usually I forgot what it's called, but here at this line, that's where the cells would align. So you can imagine by changing the frequency of the wave, by having two directions, you can actually form different type of cell clusters. And that's demonstrated here. So unpatterned, of course, you have a more or less homogeneous distribution of cells. If you have bidirectional ultrasound with a certain frequency, you can make little dots or spheroids. If you apply only across one side, your ultrasounds, you will actually form these fiber-like structures. And once they are formed, you can expose the gel to light, essentially forming a cross-linked hydrogel, essentially locking the cells in that configuration. And that's kind of what it looks like long term. So at day 0, you can still see there's individual cells, but by day 1, they already started to melt together, forming these myobundles or myofibers as well. And that only gets, more pronounced over time. And by doing clamping experiments, James was also able to show there's an anisotropy in mechanical properties. So they're stiffer essentially in the direction of the fibers versus orthogonal. And of course, once you culture those, they will also start expressing, They will start expressing all of the relevant markers, skeletal myosin, tropomyosin, while also having very well aligned myobundles as well. And you can see, of course, these start twitching spontaneously over time as well. Let's move a little bit further in the stiffness range. And look, I'm almost done with the scientific part of the presentation. Again, hopefully just a high-level overview today. But some work actually from my PhD that was before we've commercialized the system as well around cartilage tissue engineering. So we've looked at mechanical stimulation of cells, right, and how that affects gene expression, how that affects ECM production in the specific context of cartilage tissue engineering. And here, during my PhD, I built this little bioreactor system that is able to apply mechanical compression or even shear onto growing cartilage tissues. Here we used human-derived human chondrocytes, so cartilage cells. And first of all, we looked at gene expression. We can, in high level, see that the expression of chondrogenic markers is increased in response to mechanical loading, both shear and compression, and also surface lubricant markers such as what's called PRG4, proteoglycan 4. I see some nodding heads. I think some people might be aware of these materials. And of course, this also translates into the accumulation of ECM, right? So here we can see the immunostaining for collagen type 2 and type 1. And in articular cartilage, you want to see a lot of collagen type 2, but very little collagen type 1, right? So that's kind of the aim here. And we can see by looking at those images, also the quantification will show you that as well, is that the collagen type 2 accumulation in these histological sections is much higher in a mechanically stimulated construct, while the accumulation of collagen type 1, again, a surface marker, a marker you don't want to see, is relatively low across both. But we did observe, interestingly, a small layer of collagen type 1 on the outside of the construct. And for those of you who know cartilage cells, this is directly correlated, again, with the phenotype of the cells. Because on the outside, some of the cartilage cells, they attach as if it was tissue culture plastic. So they spread, they elongate on the outside of the construct. And these cells typically express fibrocartilage markers like collagen type 1, while the cells on the inside of the construct, they're more rounded and they express more collagen type 2. Okay, very last application. So while this is not the main focus of what we do and how we do it, some of our customers also use the material for 3D printing. So these are more the high stiffness kits. And this is just showing some basic data sets here that we can actually create a bio-ink that's shear thinning, meaning it can, extrude into these predefined geometries, and also allow cells here, for example, human fibroblasts that are derived from, I think, the dental, some dental human fibroblasts. I can't remember where exactly these are from. And they grow and they live, of course, as well. Cool. So, actually, there's one more little thing. So just a quick summary also how this differentiates from Matrigel, which is, of course, the leader in the market still. So we use a semi-synthetic system, which is much more controllable rather than a mouse-derived tumor, where, of course, the batch-to-batch properties can vary quite significantly. So the composition is much more controlled, more reproducible. We have tunable mechanical stiffness. It's more consistent. There's no active growth factors in the system, meaning that allows customers and users of the system to add their own growth factors and control the growth conditions much better. It's much quicker to use. The handling is at room temperature, and it's also more scalable. And again, this is some mechanical data sets, but what I wanted to point out here is that the base membrane extracts of Matrigel, they're typically much softer, and there's also significant differences between individual lots of the material, because again, these are inherently tied to the biochemical composition of the material. Last data sets, I promise. Just some high-level comparison of the growth patterns of some, commonly used cell types that we have in the lab, and also other, scientists worldwide use as well. You can see, for example, on the left-hand side, these are MCF7 spheroids, which we've looked at a few times already today. They, in LunaGel over long-term culture, they form, these fairly rounded spheroids. They're easy to quantify in size and so on, and expression can be quantified. While in Matrigel, you have a much more undefined growth pattern. It often forms these surface adherent layers. They're not actually spheroids like that. You might find some spheroids inside of the material. And these are, again, our HUVEC cells. And again, in Matrigel, some batches, sometimes it works, sometimes it doesn't. In this particular experiment, it didn't form capillary systems or structures, while in our material it did. Awesome. Look, maybe before I go on to opportunities to collaboration, do you have any specific questions around the science? I didn't go much into organoids today. I really wanted to keep it high level. We also do have organoid applications, for example, intestinal organoids. We also have cardiac organoids, iPSC derived.